Hello and welcome. I am Dr. Anchal Tandon and we have done our neoplasia topics up till invasion and metastasis. Today we will be going from evasion of immune surveillance, and this is can also be very important for the postgraduate students, for the people preparing for NEET PG, and very important for people preparing for NEET super specialty in the pathology field. So this can also come in the short notes as immunotherapy, that the basics of immunotherapy lies in this topic, and it is a very important topic. So now let's study about it. Long one in the long run of the holy grains of oncology, the promise of therapies that enables the host immune system to recognize and destroy the cancer cell is finally coming to fruition, and largely due to clearer understanding of the ways by which the cancer evade the human host response. So what we are going to study is the cancer cell is attacked by the T cells. So we'll study how these cancer cells are attacked by the T cells. Then further on we'll study how the cells are trying to escape, the tumor cells are escaping these mechanisms of killing by by these immune cells. And then we'll study about the immune therapies that can help these immune cells to kill these tumor cells. So this is under three headings. So now let's study first about the scientists that have given their roles in this thing. That is the Paul Ehrlich. This person, he first conceived the idea that tumor cells can be recognized as foreign and eliminated by the immune system. So this person was the first person who said that the cancer cells are not acting as a self-antigen, but these are acting as a foreign antigen. So they can be detected by the immune cell and they can be killed by the immune cells. And then subsequently there were two more scientists, that were the Louis Thomas, and then there was Macfarlane Burnett, who formalized the concept. They coined the term, that is the immune surveillance, which implies the normal function of immune system is to scan complete body, detect the malignant cell, and then they destroy them. Okay. Then the idea has been supported by many observations have been done, and then it has been supported. Then we'll study how this is happening is the tumor antigens. The malignant tumors express various type of antigens that help them recognize. So first we'll understand how it is being recognized. So there are these cells that are CD8 cytotoxic T cells. They play one of the major role in detecting the cancer cells and killing them. So in normal situations, a normal cell is having a ID card, that is its MHC class 1 protein. The function of it is to show the antigens that the cell is carrying to the CD8 T cells, and then this CD8 T cell determines that these are self antigens or these are non-self antigens. And how it is being recognized? Because these antigens in earlier life have been exposed to the body's immune system and a tolerance has been developed to them. But if any new antigen will be there for which the tolerance has not been developed, it will be detected as a foreign antigen. So what happens in the tumor? There will be mutated proteins. These mutated proteins, how these mutated proteins have happened? Because there is a lot of mutations. Some mutations are leading to driver mutations. These driver mutations are leading to molecular hallmark, the 8 molecular hallmarks that are very much needed for any cell to develop into a tumor cells. The rest, for every driver mutation, there is a lot of passenger mutations that are happening, and those passenger mutations will eventually lead to expression of a lot of antigens over the tumor cells. This expression of antigens will actually help the CD8 T cells to recognize these tumor cells as foreign, and the expression of these self antigens to CD8 cells will lead to a lot of immune response by the CD8 T cells. Another thing is the tumor cells. This antigen was normally present in the body, but still its overexpression is leading to immune response. So it discusses the overexpression or aberrant expression. For aberrant expression, it means there are some antigens that were in the stem cell, but now they don't express in adults, and their expression can also lead to development of immune response by CD8 T cells. The example for this is the overexpression of tyrosinases. This is normally present in melanocytes, but it is present in very less amount. So although it is still in a hypothesis that why is a CD8 T cell developing an immune response to tyrosinase when it is already a normal component, but it is being suggested that because it is in a very low amount, when it will be in very high amount, the immune response is developed. And then the example for aberrant expression is cancer antigen, cancer testis antigen. So, and there is also a melanocyte antigen gene. So these are the antigens that are not expressed in adult life but present in the germ cell life, and so later on, if they are expressed, the T cell response is evoked to it. Then there are oncogenic viruses that we have already studied, that HPV, EBNA virus. So these viral proteins, if the MHC1 presents them to CD8 cells, this will also lead to development of the malignancies. So there is the same things they have been discussed in the headings. The first one is neoantigen presentation. They have discussed neoantigen presentation, that new antigens that have been developed by the cancer cells are being presented to the T cells, are leading to immune response. Then overexpressed and aberrantly expressed, that I have already discussed, that is the melanoma antigen genes. Then tumor antigens produced by oncogenic virus that we have already discussed. These are due to, this is due to EBV, that can be due to EBV, HPV or DNA viruses, any virus. Then we will study how it is happening. So now we are studying how normally immune system is reacting to the tumor cells. So the principal immune mechanisms of tumor eradication is killing of the tumor cell by cytotoxic T cells specific for the tumor antigens. So it was seen that when the serum of the cancer patients were taken, there were a lot of antibodies developed to lot of antigens that were on the tumor cells, but they were not very effectively killing them. Okay, so why is that? See, it is seen. Although sera from cancer patients may contain antibodies that recognizes the tumors, there is little evidence that it has any protective role. Okay, so that cytotoxic T cell response against tumor was initiated by recognizing of tumor antigens or host antigen presenting cells. So now we can see that suppose there is a tumor. This tumor, some part of it has died. Not this one. Some part has died. So whenever a cell is dying, it will be phagocytosed. When it is phagocytosed, a dendritic cell has taken it and now it has started to express. Now, if you remember, we have studied in immunity chapter, dendritic cells in normal situations express MHC2 and they present to CD4 T cells. These CD4 T cells, what this will lead to? Activation of the CD8 cells and B cells for the further prevention of infection. So this is mainly what happens in the type of infections. But what is happening? This dendritic cell, normally it had to express to the CD4 cells, but there is cross presentation. This dendritic cells, and because of the cytotoxic T cell, it secretes some cytokines that leads to expression of MHC class 1. This MHC class 1 directly binds to CD8 cytotoxic T cell, causing the presentation of this antigen of the tumor, and then this cytotoxic T cells will go to the tumor and cause the killing of the tumor cells. Okay, so now it says the activation of antigen specific cytotoxic T cells also require a co-stimulatory molecule which are upregulated on the antigen presenting cells, presumably by danger signals and release from damaged or necrotic tumor cells. Okay, so they say that this antigen presenting cells are also having some co-stimulatory signals that are activating cytotoxic T cells, that is only being activated due to the DAMPs. These DAMPs are damaged or necrotic tumor cells. Then we see this cytotoxic T cell capabilities were studied, and it was found that these are very efficient in killing the tumor cells. So a lot of immunotherapies are being developed by these are engineered. They are developing engineered cytotoxic T cells. These are having that express chimeric antigen receptors, so they can become very lineage specific by engineering, and they can kill a very specific type of, like CAR T cell specific for B cell antigens have been developed. They are very highly active for B cell tumors, but also annihilates the normal B. So it is also killing the normal B cells and release inflammatory cytokines and cause substantial morbidity and even death of the patients. Apart from the cytotoxic CD8 T cell, it was seen the tumor cells that are having infiltration by the CD4 T cells, and these patients, especially the T helper type 1 tumor infiltration, were having the better prognosis, and especially this has been seen studied in the colorectal carcinoma. And in many experiments it is seen that even natural killer cells and activated macrophages are capable of killing the tumor cells, but they are only capable after the interleukin 2 and interleukin 15 causes the activation. So this natural killer cells, they cause lysis of the tumors, and this is very much non-immunogenic. It doesn't depend on the cytotoxic CD8 T cells or MHC class 1 protein. These tumors that are killed by natural killer cells may be so not immunogenic that they might not induce the CD8 T cells to kill them, but natural killer cells can kill them. Then further, the macrophages are activated by the interferon gamma that helps in killing the tumor cells. Now we will study what is the mechanism by which the immune evasion by the cancer cells, how these cancer cells are preventing themselves to be killed by this immune system. So what they are doing, there were many cancer cells. We have studied it as very much subpopulation. Some are having different phenotypes, some are having different phenotype upon multiple divisions. So there are the ones that were having a lot of antigens expression were killed by the CD8 cytotoxic T cells, but the ones that were not having the antigen expression, the antigen negative variants, were naturally selected and they outgrow. So one thing, how they have escaped, some cells have escaped or some part of tumor has escaped the immune response, is because some of it is having are antigen negative. Okay, so this first diagram that is depicted, it shows that how normally it does. There are tumor antigens, there is MHC, the MHC is showing the antigen to the cytotoxic CD8 T cell. But if there is, in the condition, there can be antigen loss. The antigen, the pink that you can see, has been lost. So there is no antigen expression. So no immune system is not able to recognize it. The another possible thing is it is not having the MHC. So the MHC is only lost. So in this situation, due to loss of MHC1, it is not able to express itself to the it is not expressing itself to the cytotoxic T cells. Then there is production of immunosuppressive proteins or expression of inhibitory cell surface proteins. So now what is happening? There is presence of antigen, there is presence of MHC, but there are some inhibitory receptors. This is leading to inhibition of T cell activation. So this thing we will study in detail, what it is saying, how it is inhibiting the T cell activation. This it is doing by upregulating the negative regulatory checkpoints. This is very, very important, and it is asked in different ways in multiple exams. So what it is doing, there is CTLA4 binds to and removes its ligands and the B7 molecules from APC, thus reducing the engagement of activating co-stimulatory receptor CD28. So we can study, we'll see this in diagram. In normal situation, for an activation, what is needed? The MHC will bind to cytotoxic T cell that we have already seen, plus there will be a co-stimulatory signal, that will be when CD28 will bind to the B7. But what is happening? CD28 is not able to bind to the B7 due to this CTLA4 overexpression. So the tumor cell is causing CTLA4 overexpression. So this overexpression is leading to binding of the B7 to CTLA4, and thus the co-stimulatory signal that was required to CD28 is not being provided, and hence this T cell will be inactivated and it will be of no use. And in the similar way, what we are doing, we are using now for development of immunotherapies to escape this thing. We have developed a anti-CTLA4 antibody. So this T cell, this the tumor cell is causing overexpression of CTLA4. So to avoid that thing from preventing the co-stimulatory signal, we have developed an anti-CTLA4 antibody that will bind to this overexpressed CTLA4, and so the co-stimulatory signal will not be hindered and this T cell will be activated, and this T cell will be capable of killing the tumor cells. And the another thing what it was doing is, the tumor cell is doing is overexpression of PDL1 ligand, and this is leading to, because the overexpression of PDL1 ligand, this is causing, this causes whenever the cytotoxic T cells will come to the tumor cells. The tumor cells have high amount of PDL1 ligand. This will bind to the programmed death receptor in the cytotoxic T cell and will cause the programmed cell death of the cytotoxic T cell, and the tumor will not be killed. This was happening normally in the tumor conditions, how it was escaping. But now what we are doing, we are using the antibodies that is anti-PD1 ligand and anti-PD1. This, what this is causing, this is preventing the tumor cells highly express PD1 ligand from binding to programmed death receptor and preventing the death of cytotoxic CD8 T cells, and then it is that is making it capable of causing the lysis of the tumor cells. So these both mechanisms are, these drugs, this anti-CTLA and anti-PD1 ligand and anti-PD1 are helping the immune system to fight the tumor cell and escape, and preventing the tumors to getting escaped from these immune cells. Apart from these all situations, they are also secreting, the tumor cells are also secreting immunosuppressive factors that are TGF beta, that are secreted in large quantities by the tumor cells and is a potent immunosuppressants. Apart from these, it also secreting interleukin 10, prostaglandin E2 and certain metabolites of the tryptophan and VEGF that are inhibiting the, so these all are actually preventing the T cell from exiting the vasculature and from going to the tumor bed. So these T cells are not being able to reach the tumor beds, and then there is induction of regulatory T cell or Tregs. So now the Tregs have function to regulate the immunity, and now they've started, these tumor cells are causing the induction of regulatory T cells only. So tumor produces the factors that favors the development of immunosuppressive T cells have been developed now. Okay, so now we come to what of this tumor new antigens, what knowledge of it can be helpful for us in future determining the therapies. So tumor, how we can determine that these tumor cells will be cleared by some specific immunotherapies. So the tumor cells that are having mismatch repair gene mutations, that are having multiple mutations, they are rapidly acquiring mutations that are leading to more passenger mutations, change in phenotypes, these are more effectively killed because there are more antigen expressions. It is seen that if you are giving PDL1 inhibitors and you are giving CTLA4 inhibitors, not all the tumors are effectively killed. Only 25 to 40% of the tumors are effectively killed by these mechanisms, because there are other mechanisms also in which these tumor cells are escaping the immunity. So we can find out if the tumor will be effectively killed by this therapy by, we can see that if the tumor cells that are, if the tumor cell is having a lot of CD8 positive T cells that are PD1 positive or do not have a exhausted PD1 positive CD8 T cell infiltrate. investigators are currently studying which biomarker will predict the responsiveness for different checkpoint inhibitor. Then we have also seen that if we are doing combination of both, like anti-CTLA4 with PD-L1 inhibitors, it is giving a better response. And this is also seen in melanoma. It is very effective in melanoma treatment. The most common toxicities that have been seen. So if you can see that what PD1 and PD-L1 inhibitor and PD-1 inhibitors are doing, this is preventing the cytotoxic T cell from being killed by the tumor cell, but it is also preventing the cytotoxic T cell from being killed from anywhere. So this will lead to increased life of CD8 cell. This will lead to autoimmune system symptoms and inflammatory damage to the other organ also. The wide range of organs may be affected. This includes colon, lung, endocrine organs, heart, skin, and so this can be a little double-edged sword. So here we complete our evasion of immune surveillance. So thank you so much, and also do like and subscribe the videos so that we can get new updates.